know someone who has cancer or has had cancer? So that's pretty much every hand in the room. Okay, so part of what we'll talk about today is cancer as well, because cancer is a result of things not dividing correctly. So we'll talk about that. But in addition to talking about cancer, we're going to talk about cell division. So there's that term mitosis that will come up, and then the cell cycle and what that looks like. But first things first, you need to just know what cell division is. So it's when a cell divides. I know, that's really hard to figure out. When a cell reproduces itself, we call that cell division, and the original cell is called the parent cell. The two cells that are resulting from that are both called the daughter cells. So neither one is a parent and then a daughter. The new ones are both daughter cells, even though one was technically there originally. So that's just some biology terminology there. Here's an example of a yeast cell reproducing. Ooh, it uses asexual reproduction. So sexual versus asexual reproduction, you need to know the differences between those. Asexual reproduction means you have a identical copy of the DNA, so the cells are identical to each other. Asexual reproduction only requires one organism, one cell. Sexual reproduction requires that two different cells combine in order to get a complete copy of DNA. So you need usually two different organisms or two different cells, and it's not an identical copy. So with sexual reproduction, you have genetic variation that takes place. With asexual reproduction, you have an identical copy. So that's another difference between the two. You should also recognize examples of sexual versus asexual reproduction. So yeast budding, like in this picture here, this is asexual reproduction. Yeast reproduces by making exact copies of itself. A lot of plants can reproduce asexually. So if you can take a plant and cut a piece off and then put it in the ground and it will grow a new plant, that's asexual reproduction. Sometimes that's called a cutting or a slipping. If you know someone who gardens, you may have known those terms already. There are a lot of other plants that can do that too. Roses are an example. So you can take a rose off of a rose bush, stick it into a potato, which helps give it some support and nutrients, and then put the potato in the ground, and you can usually get a whole new rose bush. There you go, if you didn't know that, okay? But what you're probably most familiar with plants is the sexual reproduction portion of it, which is seeds. So a seed forms when the pollen from one plant pollinates or fertilizes the egg on another plant, and then the seeds form around that fertilized embryo, and then when you plant the seed, then it grows to a new plant. Okay, that's sexual reproduction because that's genetic variation. It took two different organisms to get that. Which type of reproduction would clone fit under? Sexual or asexual reproduction? Asexual reproduction. So that's when, if you think of a clone, technically, asexual reproduction is kind of like cloning because you get an identical copy. Okay, it's another good term to know where to put. Okay. Is your family an example of sexual or asexual reproduction? Sexual reproduction, because that's how humans reproduce. No, there is no asexual human reproduction yet. We have not mastered cloning. Okay. The next thing you need to know, this is on the next page, so 8.2, is that prokaryotes reproduce by binary fission. So you should be able to describe the process of binary fission, and you should know that prokaryotes are what use that to divide and to reproduce. So binary fission is a type of asexual reproduction because it's something that single-celled organisms can do, and it literally means dividing in half. Okay, so cutting yourself in half. So this is the process of binary fission. So you have a single cell with one copy of its DNA, then it makes another copy, so you have two copies of DNA. The cell gets longer, so it spreads apart and gets wider and wider until it's long enough that it can pinch off in the middle, so the middle starts to squeeze together, and then it completely breaks apart and you have two cells. So this is just a specific type of asexual reproduction, but it's not that complicated. So when I say describe it, these are the kinds of steps that I'm talking about, the process of what happens. There's not really any technical terms that you have to know to describe it. Ben?
So is it duplicating all the organelles?
So it duplicates organelles usually before this point happens, but yes, you have to have enough organelles in both of the new cells so that they can function correctly. Okay, and then after this happens, these cells are probably going to be smaller. Well, not probably. They will be smaller than the original parent cell. So these cells will have to make more organelles and get bigger so that they are the right size again. So they do some growing after they finish dividing. Okay. Why is binary fission asexual reproduction?
It only involves a single cell.
Okay, it only involves a single cell. Good. What else? The DNA is identical, right? So those are two different characteristics you can use to help answer that. Okay. Let's look at the next page. Eukaryotic division is not as simple as binary fission. There's quite a bit more involved. There are more processes. So just related to the DNA itself, these are terms that you need to know off of this page. So there's chromatin and chromosomes. There are sister chromatids and centromeres. So chromatin, we'll talk about that one first. This is DNA that's not wound up, so it's loose inside the nucleus. This is the state DNA is in most of the time. So most of the time in your cells right now, it's chromatin. It's not a chromosome. So if you were going to draw a picture of the chromatin, you would take your pen or pencil, put it on the paper, and squiggle it around. That would be chromatin because it looks kind of like a ball of string, okay, or a pile of yarn. What you're used to thinking of as DNA is probably a structure that looks like this, kind of like an X. This is a chromosome. These only exist when the cell is getting ready to divide after the DNA has been duplicated. So the chromosomes usually exist singly on their own. So you have 46 chromosomes in your cells. That's humans. So 46 chromosomes or 23 pairs. When your cell gets ready to divide, it has to duplicate that. So then you go from 46 to 92, and they exist in sister chromatids. So the sister chromatid is when this DNA has been duplicated, you have your original copy, and then you have a second copy. So the sister chromatids only exist if DNA has been duplicated and is existing with its duplicated part right next to each other. Those are sister chromatids. So when your cells get ready to divide, like you're making new skin cells, or you're growing hair, or you're growing nails, so this happens all the time, it's a constant process, you have sister chromatids in those cells before they divide. Okay, they're connected in the middle at a particular set of proteins called a centromere. So chromatin versus chromosomes versus sister chromatids versus the centromeres. Questions about those different terms? Yeah.
What if it doesn't divide?
Ooh, if it doesn't divide, then we have issues and errors. Or if it divides incorrectly, then there are issues and errors. We'll talk about some examples of that. Okay. But it's possible for this to not split apart correctly, and that can cause different problems. It's possible for the cell to duplicate the DNA the wrong way, so then it can't ever divide like it's supposed to. So we'll talk about what that looks like, okay? Or at least some examples of what it looks like. On the next page, this figure is your friend. You want to know this figure, like the parts and the order and what happens in each part, okay? So this is called the cell cycle. Most of the cell cycle, if you look on the outside of the circle here, is interphase. So during G1 and S and G2, the cell is all in interphase. So you would need to know that interphase has those three parts. You should know kind of what those three parts are. So G1 is a first gap, S is the DNA synthesis, G2 is considered a second gap. After those, then comes the M phase, which is also called the mitotic phase. That's when mitosis and cytokinesis take place. So remember we asked at the beginning of class what mitosis is? This is how eukaryotic cells duplicate themselves. Okay, so this is only for eukaryotic cells, not for prokaryotic cells. Mitosis is the process that it describes how the organelles kind of get split to either side, how the DNA gets pulled to either side. Cytokinesis is when the cells actually break apart into two new cells. So cyto meaning cell, and how they divide. Okay, so you need to know the cell cycle. You need to know what happens in each phase. So in interphase, primarily the cell is growing. In G1, it's growing so that it's up to a normal sized cell. In S, that's when your DNA is copied. In G2, the cell's growing again so that it can be ready to divide. So Ben had asked, do your organelles get duplicated or not? And this is when in G2, organelles would be duplicated. Okay, so they'd be doubled there. And that's interphase. There is a few more pieces of information on the page about what happens in each. You should pull some examples from those. Give me just a second, Lucas.